A simple greenhouse garden in 19th century Austria was the birthplace of modern genetics. The gardener was an Augustinian monk named Gregor Mendel. Mendel worked tirelessly with his pea plants to crossbreed the characteristics of each and to determine which traits would be passed from one generation to the next. From the data he gathered, Mendel's analysis showed that patterns of inheritance were mathematically predictable. The seeds of inquiry that were sown in a modest garden led Gregor Mendel to formulate his laws of inheritance and to lay a foundation for the science of heredity and genetics. In this program, you'll review some early theories of inheritance and then study Gregor Mendel's principles of heredity. The vocabulary of modern genetics will be used as you explore the results of a monohybrid cross for a particular genetic trait and look at the importance of doing test crosses. You'll also do some math in this program. You'll learn how probability can be used to predict particular genotypes and phenotypes appearing in a generation. Inherited traits have long intrigued scientists, especially when the characteristics do not appear in the parents. Heredity is the term applied to this passing on of traits. Aristotle's theory of pangenesis claimed that tiny pan genes came from the cell, from all parts of the body. These combined particles were given to the unborn child, so that all characteristics could be inherited. Today, we use the term gene. It's the factor that does come from each parent to affect heredity. The word is derived from Aristotle's language of Greek, and it means to give birth to. In the 17th and 18th centuries, two Dutch theories about heredity emerged and clashed. The scientists who sided with Anton van Leeuwenhoek, the inventor of the microscope, claimed to see a miniature human within the sperm. René de Graaf noted that aphids can reproduce parthenogenically. That is, the female eggs can develop without being fertilized by sperm. This led to the belief that the egg held the homunculus, or miniature baby, while the semen simply stimulated its growth. These miniaturization theories were challenged when Caspar Wolff, a German physiologist, noted that specialized organs arise out of unspecialized tissues in a growing plant. He later found that the cells of human embryos also become differentiated during growth. Clearly, a tiny human was not within the fertilized egg. In the early 19th century, biologists observing plants established the blending theory. That is, that the materials for inheritance come from both parents and are mixed at fertilization. But Gregor Mendel's model of inheritance rejected the homunculus and the blending concepts. Mendel showed that the breeding of tall pea plants with short or dwarf pea plants did not result in plants that were of medium height. Rather, a fixed ratio of tall and dwarf plants resulted. By studying other characteristics, Mendel was able to show that mixes of characteristics did not blend, but retained their identity. He demonstrated pairs of characteristics combined and sorted themselves according to fixed, simple rules.
In the early 19th century, biologists believed that blending was responsible for pink offspring when you crossed red and white snapdragons. We now understand that this is not the case for the traits that Mendel studied. But how did he figure that out?
The reproductive structures of the pea plant are contained in each flower on the plant. They are the stamens and the pistil. A flower self-pollinates, and pollen from the stamen is transferred by wind or insects to the sticky surface on the pistil. A pollen tube develops and grows down the pistil to the egg cell in the ovary. The sperm nucleus in the pollen tube combines with and fertilizes the egg cell. Mendel had to ensure that he could control the parentage of seeds. He wrapped plants that he wanted to self-fertilize so that they wouldn't be pollinated and fertilized by other plants. He clipped off the anthers of flowers on other plants so he could pollinate plants himself with the pollen from other specific plants. This is cross-pollination. It results in the fertilization of the egg cell of one plant by the sperm nucleus from another plant.
I must make this clear for the Society of Natural History. These plants all had terminal flowers, flowers that grow from the ends of the stem. After they had been self-fertilized, they produced plants that had terminal flowers. I called these plants my P1 generation, plants that I knew were true breeding. These plants produce flowers that grow from the side of the stem, actual flowers. After these plants had been self-fertilized, they all produced plants that had actual flowers. These plants are true breeding. I decided to make my first cross with true breeding plants. Cross-fertilized true breeding plants for terminal flower with true breeding plants for actual flowers. All the flowers produced in the F1 or first filial generation had axial flowers. I wondered where the terminal factor went. I decided to cross the F1 plants. The F1 plants were not true breeding. That is, they did not produce offspring like themselves. Of the plants of the F2 or second filial generation, 3/4 produced axial flowers and 1/4 produced terminal flowers. The factor for terminal flowers had come back. Curious to know more, I let each plant of the F2 generation self-fertilize. Of the plants produced, 1/4 were true breeding for axial flowers, 1/2 were not true breeding, or hybrid, but produced axial flowers, and 1/4 were true breeding for terminal flowers. Clearly, the hybrid plants contained factors for both characteristics, but produced only axial flowers. The factor for terminal flowers had not disappeared. It was only masked. When it combined with a plant that had a similar factor, it produced offspring that had terminal flowers. To explain these results, I believe that the hereditary factors must occur in pairs. These factors must separate in the parent plant, and then each parent must give one factor randomly to the next generation. I will call these factors either dominant or recessive. The dominant factor masks the recessive factor. In this case, the true breeding axial plants have two dominant factors, capital A, capital A. The true breeding terminal plants have two recessive factors, small a, so the trait can show. The hybrid plant has one dominant factor, capital A, and one recessive factor, small a, but only the dominant factor, axial flower, shows.
Okay, here's a list of terms in modern genetics. Let's see how well you do defining them as we review the work of Mendel. Just to make it interesting, if you define it correctly, I'll give you the card and we'll tally up the number of cards at the end.
Okay, well, what Mendel first called factors are called genes now.
Okay. Define genes.
Genes are areas on the chromosomes that define our traits. And chromosomes are long strands of DNA.
Good. Yes.
We learned about chromosomes when we studied meiosis. Meiosis is the separation of chromosomes when reproductive cells divide to form gametes.
That's true. Well done. Okay, can you define gamete?
Well, it's what you get when there is meiosis.
It's actually what you get after meiosis. Gametes are a haploid sex cell with a complete set of chromosomes, unlike a somatic or body cell with two complete sets of chromosomes.
Excellent.
I'll take fertilization. That'd be when two gametes combine, the sperm cell and the egg cell. So you get a fully ploid cell.
Yeah, it's actually called diploid, but I'll give you the card anyway.
Thanks.
Let's see. Well, I know homo means one and the same, and zygous probably refers to the zygote we were talking about earlier. So homozygous means that an individual has all the same genes?
Close. You need to deal with alleles first. Okay, I get that card. Alleles, two or more forms of a gene, a specific location on a chromosome. Draw a pair of homologous chromosomes. We'll put the location for the gene flower position here. There are two alleles for this trait. Axial position and a terminal position. Mendel found that the axial allele is dominant, so we give that a capital letter A. And the terminal position allele is recessive, so we write small a. Now there would be other pairs of alleles on other homologous chromosomes, maybe for flower color or stem length. In each individual of a species, the genes are ordered, strung out along a chromosome, and on homologous chromosomes, that order is the same. Like that. Would you care to rephrase your definition for homozygous?
Okay. Homozygous is when an individual has the same set of alleles for a trait, dominant or recessive.
Right. If we self-fertilize them, what kind of breeders would they be?
Well, they would probably be true breeders, giving the same traits to every gamete.
Perfect. Why don't you try one, Nicole?
Well, I could guess that heterozygous means different in the zygote. So that'd be one dominant allele and one recessive allele.
Perfect. Two left. Let me give you a clue. Pheno is a Greek combining word meaning shining or appearing.
Shining or appearing. So the phenotype would be the appearance of a type or a trait. Then genotype would be the actual allele an individual has. So the genotype, capital A, small a, would appear as the phenotype, the axial flowered pea plants, because the axial allele is dominant.
Good job.
I got 3.
I got 6. I have 1. The sperm nucleus carried by the pollen and the egg cell are gametes formed by meiosis. These gametes are haploid. Since each gamete can only carry one allele for any trait, the alleles must have segregated during meiosis or gametogenesis. When the haploid sperm nucleus and egg cell meet during fertilization, the complementary alleles match up, potentially in new combinations, and the diploid condition is restored in the offspring. I've brought you the harvest of a monohybrid cross between two heterozygous plants. Now, mono means just one. So in this case, we're looking at just one genetic trait. And the trait determines whether or not these peas will be wrinkled or round. Round is dominant. What are the genotypes of the heterozygous parents?
If round is dominant, they use the capital of the first letter in the word, so capital R, then small r to show recessive and therefore heterozygous.
Great. I'm going to give each of you a beaker, and here are 2 plates. What you need to do now is to separate the peas into round and wrinkled piles, and then count all the peas in each pile. You can determine the ratio of each seed type.
Okay. The round ones here, and I'll put the wrinkled ones here.
Okay.
When chromosomes are separating in the first meiotic division, each pair of alleles split, since they are carried on homologous chromosomes. We say that the individual alleles segregate. Mendel called this the principle of segregation. It became known as Mendel's first law, the law of segregation. You finished your calculations?
Yeah. We came up with 657 peas altogether.
We counted 156 wrinkled peas and 501 round peas.
It's a lot.
The ratios were 156 over 657 for the wrinkled, which came out to 0.237. And for the round, it was 501 over 657, which came out to 0.763.
That'd be about 1/4 wrinkled and 3/4 round peas.
Okay, good. Let's use a Punnett square to predict the probable ratios of the seed types in the offspring. Do you remember the genotypes of the parent plants?
Yes, capital R, small r.
Okay, during meiosis, those would separate into gametes. And the gametes are what we write on the outside of the Punnett square. So we'd have capital R and a small r for one parent. And then we have another capital R down here and a small r. Now, just as it would be at fertilization, the way to figure out a Punnett square is we combine the gametes. So what's the genotype of the zygote that would be here, Cam?
Big R, big R.
Right. Thanks, Nicole. Okay, what about in the square beside it? This one.
Capital R, small r.
Good. Would it be the same for this one, too?
Yes.
Good. Thanks. And what about the last square?
2 little r's.
Good. Okay, that is the Punnett square completed. What does it tell us?
It tells us all the genotypes of the next generation. While we were separating the peas, we looked at the phenotypes and the probability of their appearance. 3 squares are round, and the last square tells us that some will be wrinkled.
How does that compare with your data?
Well, your data shows that about 1/4 will be wrinkled and about 3/4 will be round. So it clearly agrees with the Punnett square.
That's fine for the phenotypic ratios. What about the genotypic ratios?
Well, 1 out of 4 have the 2 dominant alleles. 2 out of 4 are heterozygous, and 1 out of 4 has the 2 recessive alleles. That's a 1 to 2 to 1 genotypic ratio.
Absolutely. Heredity is like a dance of generations, ensuring tremendous variety of inherited traits, even among siblings. Traits can reappear 2 or 3 generations later if the same recessive trait is brought in by a new partner. We need to explore the rules of probability that make the Punnett square such a valuable predictive tool. So you can understand this. We say that alleles segregate randomly during gametogenesis, and we can predict the probability of random events using the rules of probability. This coin has 2 sides. What's the probability that it will come up heads on its next toss?
Oh, that's easy. 1 out of 2, or 1/2.
And the probability it will come up tails?
The other half of the time.
Good. Now what's the probability that will either come up heads or tails?
1/2 plus 1/2 equals all the time. 1.
Good. Probabilities can also be expressed as decimals. 0.50 plus 0.50 equals 1. Here's a tougher question. What's the probability both these coins will come up heads?
Well, for each coin, the probability is 1/2.
Yes, but there are 4 different possibilities. You could have 2 heads, 2 tails, left tail, left head, right tail, or right head.
Exactly. We can multiply the probabilities of the independent events to find out the probabilities of the simultaneous events. So what's the probability that both coins will come up heads?
1/4 or 0.25.
Excellent.
This Punnett square shows the possible combinations of heads and tails when 2 coins are tossed. Segregation of alleles during gametogenesis of a heterozygous plant is just like flipping a coin. The gamete can either get a dominant allele, shown here as capital R for round seed, or a recessive allele, shown here as small r for wrinkled seed. The other heterozygous parent offers the same 2 alleles in its gametes. The chance that the fertilized zygote will get both recessive alleles is 1 out of 4, that is 1/4 or 0.25. We can use the multiplication or product rule to get the same result. the chance that the zygote gets a recessive gamete from one parent is 1 out of 2, 1/2, or 0.50. And the chance of receiving the recessive gamete from the other parent is also 0.50. Thus, the overall probability that the two recessive alleles will come together at fertilization is 1/2 times 1/2, which equals 1/4, or in decimal terms, 0.50 times 0.50 equals 0.25. This is probability of any offspring being homozygous recessive for seed coat, provided that both parents are heterozygous. So what traits do you breed for, you personally?
Well, basically song, and although a lot of the breeders in general don't really go for the conformation, but I like to see a good-looking bird, so I sort of try to breed for conformation as well, probably secondary to song, but still it's nice to have nice big birds. One of the differences in them is that the whites are not quite as good a songbird as the yellows are, even after all of these years that they've been raising them. It takes probably about 6 generations to really fix a strain. If you don't have any whites and you're going to start out from scratch, so to speak, the only way you can get a white is from a mutation, actually. So you get a mutation, so unfortunately it's probably not a singer. It's just a canary, like. But as long as it's got the white gene in it, that's what you really want. So you cross that with your water slaggers. Then your first strain, the first offspring, you call them F1s, and they'll be good singers. They'll sing good, but they won't have the qualities of the water slaggers, all of them. So every year you keep bringing the offspring back to a pure water slagger, like the white ones, that is, because you got to keep the white strain.
Right.
And you keep improving your song that way, and within probably maybe 3 generations, you got some pretty good singers. But by 6 generations, they should be really good by that time.
Have you seen anything that just amazed you, or something that you never really expected when you were breeding?
There's in the color-bred line, there's an awful lot of different color-bred birds, and it's simply amazing the new colors that are being generated, and it's through this same old thing of borrowing genes and crossing them in and line breeding back and forth until you get some really nice looking birds. Probably about 50 years ago, they wanted to raise reds, and I guess they were trying to do it by color feeding. To a degree, that'll work. Some birds you can color with color, but how they really did get the reds in, they needed a gene. So without having a mutation, they went to the Venezuelan red-hooded siskin, and which of course had red, and they brought that in and mated it with yellow canaries. And the only trouble is, when you cross a different species, you're unfertile probably. But they did get fertile hens, and after about 3 generations, they started to get fertile males. And so that's the way that basically how they started out getting the reds. Today there's some mighty fine red canaries around. So you either borrow a gene from someplace in something that you can breed into the canaries, or you wait for a mutation. That's the only other way.
So how long have you been doing this?
Well, I started as a teenager, which is many years ago, and still at it.
One important tool in genetics is the test cross. Test crosses are used to determine the genotype of a particular individual. In a test cross, the individual of a known genotype is crossed with a homozygous recessive. By studying the frequencies of the phenotypes in the offspring, one can determine the genotype of the organism in question. Suppose you want to know the genotype of a pea with a round coat. Since round is dominant to wrinkled, It could be either capital R, or capital R, small r. To determine which is true, the individual is test-crossed with a homozygous recessive individual, one carrying the alleles for small r. The frequency of traits in the resulting offspring will reveal whether the plant was heterozygous or homozygous. If the unknown individual was heterozygous, half the offspring would be heterozygous round, capital R, and half would be recessive wrinkled, small r. If it was homozygous, having two dominant alleles for round coat, all the offspring will be heterozygous dominant and appear round. In this way, a test cross reveals what genotype is responsible for the phenotype. Important information any breeder needs to determine whether a dominant trait is pure or hybrid. Now that we know why test crosses are so important, I'm pleased to say that all of these plants have proven to be true breeders. In this box here, we have all homozygous tall plants, or 2 big T's. And in this box here, we have all homozygous short plants, or dwarf plants, 2 little t's.
Great! So now we have to be sure that we cross-pollinate them properly.
You get ready to remove the anthers off of one of the tall plants, while we wait for the anthers in one of the dwarf plants to mature. Then, when the pollen is ripe, we can cross-fertilize.
So, right here?
That's one.
Careful.
Good job. Here's the completed Punnett square of our project.
We predict that all the offspring will be heterozygous for plant height. Because the tall allele is dominant, all the F1 generation will have the same phenotype. They'll all be tall plants.
You're right. Mendel's precise attention to data collection, his curiosity, and his careful analysis of the numbers and ratios led him to the discovery of the principles of inheritance. And his groundbreaking work wasn't verified until the microscope revealed chromosomes within the nucleus of a cell, and that was many years after Mendel's death. We now know that the system of paired alleles and the random segregation of those alleles in gametogenesis ensure genetic variation within a gene pool. They also allow for a successful mutation in one parent to be passed on to the next generation with predictable ratios. In a square dance, partners pair and separate and combine with others to form new squares. In much the same way, gametes join in fertilization in a dance of genetic recombination that carries from generation to generation.
Make a left-hand star, 1, 2, 3, 4, 5, back for the race. You join your hands and turn the left all the way. And the ladies take, left-hand star you go. And back by the race you go. Form your running slant, left and right. Ladies take hold. Circle forward all the way wherever you go. Ladies change straight up. Left hand start go. And back to the right.